Put your hands in soil and something happens. Every gardener knows this, the shift that arrives within minutes of bare hands entering earth. An alertness that was not present before. A clarity that indoor air did not provide. A mood change that feels biochemical, because it is biochemical. The experience is so consistent across gardeners, across cultures, across decades of anecdotal reports, that it demands a mechanism. If you garden, you have felt it. The specific difference between the first minute indoors and the first minute with your hands in earth. The shift is not gradual, it arrives. Something in your body responds to the contact in a way that no indoor activity replicates. Not typing, not cooking, not holding a book, not any other hand-intensive task. The hands in soil produce a response. The hands on a keyboard do not. The question is, what crosses the boundary between your skin and the earth to produce it? The mechanism is not one thing. It is 6 things. 6 independent physical events that occur simultaneously when skin contacts soil, each crossing the barrier between your body and the earth through a different pathway, each producing a measurable change in a different system. The 6 mechanisms operate through different physics, target different organs, and produce different effects. But they activate simultaneously the moment bare skin enters soil, and the combination produces the integrated response the gardener experiences as waking up. Barrier is where the physics begins, because the boundary between your hand and the soil is not a wall, it is a membrane. And understanding what crosses it requires understanding what it is made of. Stratum corneum, the outermost layer of your skin, is approximately 10 to 15 micrometers thick on the hands. 15 micrometers, thinner than a sheet of paper, thinner than the diameter of a human hair. This is the entire barrier between your internal chemistry and the chemistry of the soil. It is composed of dead keratinocytes embedded in a lipid matrix, a brick and mortar structure where the bricks are flattened dead cells and the mortar is a mixture of ceramides, cholesterol, and fatty acids. This structure is a selective barrier. Small molecules, those with molecular weight below approximately 500 Daltons and sufficient lipophilicity, can penetrate through the lipid mortar by passive diffusion, following concentration gradients from high to low. Barrier selects by size, by charge, and by solubility. It does not select by intent. Anything that fits through the mortar crosses the barrier whether you invited it or not. Hands have a specific vulnerability that the rest of your skin does not share to an equivalent degree, microabrasions. Any person who gardens, who has pulled weeds, turned soil, handled rough stems, worked with roots, has countless micro breaks in the stratum corneum. Cuts too small to see. Abrasions from friction with soil particles. Thorn pricks that healed on the surface but left channels through the keratinocyte layer. Cracks from drying. Each microabrasion creates a direct pathway through the barrier, exposing the living epidermis to the soil's chemical and biological contents. Your gardening hands are not merely touching soil, they are open to it. What crosses first is temperature, and the body's response to it is immediate. Soil at shallow working depth, 5 to 15 centimeters, ranges from approximately 10 to 18 degrees Celsius in temperate spring and autumn. Your hand skin at rest is approximately 30 to 34 degrees. The temperature differential when your hands enter cool morning soil is 12 to 24 degrees, sufficient to activate TRPM8 cold receptors in the skin. These are the molecular sensors that respond to temperatures below approximately 25 degrees by opening an ion channel that generates a neural signal, a direct cold detection mechanism operating at the level of individual receptor proteins in the cell membrane. You feel this immediately. The first contact with cool earth, the slight shock of it, the sharpening of attention, the sense of the hands becoming more awake, that is TRPM8 activation. The cold receptors fire. The signal travels through the sensory nerves of the hand, up the arm, through the dorsal columns of the spinal cord, to the somatosensory cortex. The brain registers, the environment has changed. The hands are in something cool, complex, and alive. The sympathetic nervous system engages not the full fight-or-flight activation of a cold shower, but a gentle alerting response, a mild increase in norepinephrine that raises attention and engagement without producing stress. TRPM8 activation from cool soil produces this mild sympathetic response, a gentle alerting signal below the threshold for a cold shock response, but above the threshold for conscious awareness. The mechanism is the physics of cold water on the face. The dive reflex discussion covered the trigeminal pathway, but at a gentler intensity, sustained over a longer duration, producing a milder but more prolonged sympathetic engagement. Wake-up effect of putting hands in soil on a spring morning is partly TRPM8 activation. The temperature differential between warm skin and cool earth, triggering a neural alert that the body interprets as engagement with the physical environment. The alert persists for the duration of the contact. The cool soil does not warm to hand temperature quickly. The thermal mass of earth is sufficient to maintain the differential for the entire gardening session. The alerting signal is sustained, not transient. What crosses next is mechanical information, and the volume of it is unlike anything your hands encounter indoors. Your hands contain the highest density of mechanoreceptors in the human body, approximately 17,000 tactile units per hand. When your hands work soil, they encounter a stimulus profile that no indoor activity replicates, varying particle sizes from clay at less than 2 micrometers to sand grains at 2 millimeters, embedded stones, root fibers, moisture gradients, temperature gradients, and resistance that changes with depth, moisture content, and compaction. Each handful of soil presents a unique mechanical challenge. No two handfuls are identical. The variability is continuous and inexhaustible. The principle: the dirt versus concrete discussion described for the foot, but applied to the body's most densely innervated sensory surface. Meissner corpuscles in your fingertips detect the flutter of soil particles shifting between the fingers. Fine-grained texture discrimination at 10 to 50 Hz. These are the receptors that distinguish silk from cotton, smooth from rough, dry from damp, and soil activates them continuously because the texture changes with every movement of the hand. Merkel cells detect sustained pressure as the hand compresses earth. Force and form discrimination, telling the brain whether the soil is loose or compacted, wet or dry, filled with roots or free of them. Pacinian corpuscles detect vibration transmitted through roots and stones as the hand pushes deeper. High-frequency transient detection at 100 to 300 Hz, registering the sudden resistance of a buried rock or the snap of a root severed by finger pressure. Ruffini endings detect a stretch of skin as the hand opens and closes around clumps of earth. Grip and shear force monitoring, tracking how the soil deforms under the hand's pressure. All 4 receptor types firing simultaneously in constantly varying patterns that no manufactured surface, no keyboard, no phone screen, no steering wheel, no indoor object produces. Compare what your hands receive indoors. A phone screen, smooth, flat, room temperature, uniform. A keyboard, repetitive, identical key resistance, predictable spacing. A book, uniform texture, uniform temperature, minimal variation. A door handle, uniform metal, uniform shape, uniform temperature. Every indoor surface your hand's contact delivers a fraction of the sensory complexity that one handful of soil provides. The indoor world has impoverished the hand's sensory diet, the way concrete impoverished the foot's, reducing a high-capacity sensor array to a low-information feed that activates a fraction of the system's design capacity. Your somatosensory cortex dedicates more processing area to the hands than to any other body part. The cortical homunculus shows hands with grotesquely exaggerated representation, reflecting the disproportionate neural resources the brain allocates to processing hand input. When all 4 mechanoreceptor types in both hands fire in complex, varying patterns simultaneously, the somatosensory cortex is receiving its richest possible input, the maximum data rate the tactile system was designed to handle. Think about what that does to the rest of the brain. The mental clarity gardeners describe, the sense of being fully present, physically engaged, the rumination that stops, the worry that recedes. is partly a cortical competition effect. The somatosensory input from your hands in soil is so rich and so demanding of processing resources that the default mode network, the neural network that sustains rumination, self-referential thought, worry, and mental replay, cannot maintain its usual activity. The cortex is busy processing soil. The rumination stops because the processing resources it requires have been allocated elsewhere. If you have experienced this, the specific quieting of mental chatter that arrives 10 minutes into gardening, the sense of the mind settling into the present without effort or intention, you have experienced cortical resource reallocation. The default mode network did not quiet because you decided to be mindful. It quieted because 17,000 tactile units in each hand demanded so much processing capacity that the cortex had no bandwidth left for the recursive self-referential loops that produce anxiety, regret, and circular worry. The gardening did not calm the mind through psychology. It calmed the mind through neurological resource competition. The hands outcompeted the rumination for cortical processing time, and the hands won because their input was richer and more demanding. Indoor activities, typing, holding a phone, turning a page, stirring a cup, activate a fraction of the hand's sensory capacity. Soil activates nearly all of it. No study has measured default mode network activity specifically during soil contact. The specific application is this discussion's inference from the established principle. The principle that rich sensory input suppresses default mode network activity is documented across multiple sensory modalities. The application to hands in soil is consistent with what gardeners report, but has not been directly imaged. The inference is strong. The direct confirmation is pending. Now, what crosses the skin chemically? Because soil is not inert particulate matter. It is an electrolyte solution, and your skin is another electrolyte solution. And when two electrolyte solutions meet across a selectively permeable membrane, the physics of diffusion applies whether you know about it or not. Soil is water held between mineral particles saturated with dissolved ions. Healthy garden soil solution contains potassium at 1 to 5 millimoles per liter, calcium at 2 to 10, magnesium at 1 to 4, zinc at trace concentrations, and iron at trace to low concentrations. The concentrations vary with soil type, pH, and organic content, but the ionic character is consistent. Soil solution is an electrolyte. It conducts current. It contains dissolved minerals at concentrations that depend on the geology of the land it sits on. Clay soils holding more ions in solution, sandy soils holding fewer, organic rich soils holding the most diverse mineral profile. Interstitial fluid, the fluid beneath the stratum corneum, is also an electrolyte. At physiological concentrations your body maintains within narrow homeostatic ranges. When two electrolyte systems contact each other across a selectively permeable membrane, which is precisely what happens when your hand enters moist soil, ions migrate along concentration gradients. Fick's law diffusion. The rate of migration is proportional to the concentration gradient, the membrane area, and the time of contact. If soil potassium exceeds skin potassium, potassium migrates inward. If skin sodium exceeds soil sodium, sodium migrates outward. The exchange is passive. It requires no cellular transport, no active pumping, no biological machinery beyond a gradient and a permeable boundary, both of which exist the moment your hand touches moist earth. The microabrasions in your gardening hands accelerate this exchange. Each break in the stratum corneum is a channel that bypasses the lipid barrier entirely, allowing ionic migration at rates far higher than passive diffusion through intact skin. Hands that have been in soil regularly carry more microabrasions than hands that garden once a year. The regular gardener has a more permeable interface with the soil electrolyte. The contact adapts the exchange. Transdermal mineral absorption through soil contact has not been extensively studied in humans. Most transdermal research focuses on pharmaceutical drug delivery rather than environmental mineral exposure. The physics permits it. The concentration gradients favor it. The microabrasion pathways facilitate it. The duration of typical gardening, 30 to 60 minutes of hand-soil contact provides extended contact time for Fick's law to operate. The minerals that gardeners have traditionally attributed to eating from the garden may also be arriving through the skin of the hands that work the soil. The evidence for this specific pathway is preliminary. The physics is established, the direct human measurements are sparse. The mechanism deserves mention with that qualification. Well, and then the microbiology. Because 1 gram of healthy garden soil contains approximately 1 billion bacteria. 1 billion. And 17,000 mechanoreceptors are working that gram into every fold and crevice of your hand skin for the duration of the gardening session. Skin hosts approximately 1 trillion bacteria, 1,000 species distributed across different body sites. The hand microbiome is among the most dynamic, changing with every surface contacted and partially resetting within 24 to 48 hours. When your hands contact garden soil, the skin microbiome is exposed to a diversity of organisms it never encounters indoors. Soil bacteria, soil fungi, soil protozoa, organisms that the indoor environment has entirely eliminated from your daily microbial exposure. Hansky and colleagues published in the Proceedings of the National Academy of Sciences in 2012 a finding the series Voice requires attention to the biodiversity hypothesis of immune regulation. The diversity of environmental microbial exposure correlates directly with the robustness of immune regulation. Populations with higher environmental microbial diversity, populations that live in closer contact with natural soil environments, have lower rates of allergic and autoimmune diseases. The mechanism runs through toll-like receptors on keratinocytes and Langerhans cells in the epidermis. Innate immune receptors that detect microbial molecular patterns and calibrate the adaptive immune response accordingly. Skin exposed to diverse soil organisms is skin whose immune surveillance system is broadly calibrated, trained on a wide range of molecular patterns, producing a balanced regulatory response that distinguishes genuine threats from harmless environmental molecules. Skin exposed only to indoor microbes, human skin flora, household bacteria, sanitizer residue, is an immune system calibrated to a narrow range. The narrow calibration is associated with overreaction to harmless stimuli, pollen, dust, food proteins, the body's own tissue proteins. The immunological basis of allergic and autoimmune conditions is, in significant part, a calibration error produced by an impoverished microbial environment. The immune system is not malfunctioning. It is responding accurately to the information it has received, and the information was too narrow to produce the balanced regulatory response that broader exposure would have produced. For the person over 65, the immune calibration question is particularly consequential. Age-related immune dysregulation, the chronic low-grade inflammation called inflammation, is characterized by precisely the regulatory T-cell imbalance that the biodiversity hypothesis describes. The immune system becomes more inflammatory and less regulatory with age. Environmental microbial exposure through soil contact provides the toll-like receptor stimulation that promotes regulatory T-cell production, the cell population that suppresses inappropriate inflammation. Soil contact in older adults is not merely gardening. It is immune recalibration through the pathway the immune system was designed to use. You see, the seeding of your hand microbiome through soil contact is not permanent colonization. Most soil organisms are outcompeted by resident skin flora within 24 to 48 hours. The skin's established microbial community reasserts itself and the soil organisms decline. But the immune calibration effect, the toll-like receptor activation, the regulatory T-cell stimulation, the cytokine profile shift toward anti-inflammatory balance persists beyond the bacterial residence. The organisms triggered the immune response. The immune system processed the molecular patterns. The regulatory adjustment was made. The organisms can leave without reversing the adjustment. Each soil contact episode is an immune training event. The organisms are the teachers. The toll-like receptors are the students. The lesson: a broader molecular pattern library, a more balanced regulatory response, persists after the teachers have left. The immune system that processed 1 billion soil bacteria during a morning of gardening carries the calibration adjustment for days after the bacteria themselves have been replaced by resident flora. The training accumulates. Regular soil contact produces regular immune recalibration, a repeated broadening of the molecular pattern library that maintains the regulatory balance. The indoor environment narrows. That calibration, the immune system trained by microbial diversity it encounters only through environmental contact, producing a regulatory balance that indoor living eliminates, is the finding that turned the allergy epidemic into an input problem for me. Because it means the immune dysregulation that drives allergic and autoimmune conditions is partly an input problem, the system is not malfunctioning. It is calibrated to an impoverished environment. Broaden the input and the calibration broadens. The soil provides the input, the skin provides the pathway, and the immune system provides the response it was designed to produce when it receives the microbial diversity it evolved with. Mycobacterium vaccae, a soil bacterium present in most garden soils, enters through microabrasions in the hand skin and through inhalation of aerosolized soil particles disturbed by hand activity. Once in the body, dendritic cells in the skin and lungs present its antigens to T cells. The immune activation triggers a specific anti-inflammatory cytokine profile that crosses the blood-brain barrier and activates serotonergic neurons in the brainstem, the cluster that is the primary source of serotonin for the forebrain. Mood shift gardeners report the specific elevation that arrives within 15 to 20 minutes of bare hand soil contact and persists for hours afterward is consistent with this pathway. Lowry's work demonstrating immune-triggered serotonin production is in mice. The extrapolation to human gardening, the 15 to 20-minute mood shift gardeners consistently describe, aligns with the mechanism, but the direct measurement of serotonin elevation from soil contact in humans has not been performed. The anecdotal consistency is striking. The controlled human data is not yet there. What is established? The bacterium is present in the soil. The immune pathway to the dorsal raphe is documented in animal models, and the mood shift gardener's report follows the timeline the mechanism predicts. Your body manufactured the mood change. The soil provided the trigger. The hand provided the entry point. And the pathway depends on the absence of gloves. Glove gardening reduces the transdermal M. vaccae exposure dramatically. The rubber or fabric barrier prevents the bacterium from reaching the microabrasions where it crosses into the living epidermis. Tool-only gardening eliminates hand soil contact entirely. The mechanism requires direct skin-soil contact. The serotonin pathway begins at the bare hand, and only at the bare hand, and only at the bare hand. And then the smell. Because when your hands work soil, they warm the surface and mechanically disturb the microbial layer, releasing geosmin, a compound produced by Streptomyces bacteria in the soil, the molecule responsible for the distinctive smell of disturbed earth. Geosmin from hand-disturbed soil enters your nose at close range, 30 centimeters from the source, rather than the diffusion distance from rain falling on distant ground. Concentration at your nose when your hands are in soil is potentially an order of magnitude higher than during rain because of the proximity. The olfactory hippocampal activation is correspondingly stronger. The memory and emotion response to the smell of disturbed earth is more intense during gardening than during rain because the source is at arm's length rather than scattered across the landscape. You know this smell. It is the specific scent of turned earth, the smell that arrives the moment you push your hands into garden soil, the smell that is different from rain on pavement and different from potting mix and different from compost. The smell is geosmin, produced by Streptomyces bacteria in the soil, released when your hands disturb and warm the surface layer, arriving at your olfactory bulb through the shortest sensory pathway your brain possesses. The nose processes it before you consciously register that you're smelling anything. The hippocampus activates. If you have gardened before, if you have decades of hand-soil contact encoded in your memory, the geosmin triggers autobiographical recall through the olfactory limbic pathway. The smell of soil carries every previous gardening experience you have ever had, arriving through a pathway that bypasses conscious processing entirely. Consider what is happening simultaneously after 10 minutes of bare hand soil contact. TRPM8. Cold receptors in your hands are firing continuously from the temperature differential between warm skin and cool earth, producing sustained, mild sympathetic engagement. All 4 mechanoreceptor types are firing in complex, varying patterns, flooding the somatosensory cortex with input that suppresses default mode network rumination. Dissolved minerals from the soil electrolyte are migrating across the skin barrier along concentration gradients. 1 billion bacteria per gram are seeding the hand microbiome and activating toll-like receptors on epidermal immune cells. vaccae is crossing the skin barrier through microabrasions and initiating the serotonin pathway in the dorsal raphe nucleus. Geosmine is reaching the olfactory bulb at close range and activating hippocampal memory circuits through the fastest sensory pathway the brain possesses. 6 mechanisms, 6 different physical pathways, 6 different target systems: sympathetic nervous system, somatosensory cortex, mineral homeostasis, adaptive immunity, serotonergic mood regulation, and olfactory limbic memory. All operating simultaneously, all initiated by one action, bare hands in soil, and then the convergence I have not been able to reduce to a single mechanism, because the 6 mechanisms are independently documented, but they are not independent in their effect. The somatosensory flooding suppresses the default mode network rumination that elevates cortisol. The reduced cortisol permits the immune calibration to operate more effectively. Cortisol is immunosuppressive, and chronic cortisol elevation specifically suppresses regulatory T-cell production. The regulatory T-cells produced by soil microbe-driven toll-like receptor activation reduce the chronic low-grade inflammation that disrupts serotonin signaling in the central nervous system. The serotonin elevation from vaccae reinforces the mood shift that reduces the stress response that was suppressing the immune calibration. The TRPM8-driven sympathetic engagement increases blood flow to the hands, which increases the rate of transdermal ion exchange by widening the dermal capillaries beneath the stratum corneum. The systems feed each other. The convergence is not additive. 6 independent effects summing to a total. It is synergistic, each mechanism enhancing the conditions under which the other mechanisms operate most effectively. Each individual link in this chain is established physiology. The circular reinforcement, all 6 operating synergistically during a single gardening session, is this discussion's synthesis from the individual findings, not a measured integrated response. The synthesis is consistent with the gardening experience. The integrated measurement has not been performed. The immune system works better when cortisol is low. Cortisol is low when the default mode network is suppressed. The default mode network is suppressed when the hands are processing complex sensory input. The complex sensory input comes from the soil that is simultaneously providing the microbial diversity that calibrates the immune system. The circle closes. The 6 mechanisms are 6 entry points into one integrated response, and the integrated response is what the gardener feels as waking up. Consider what this means practically. Because the mechanisms do not require a garden, activity itself does not matter. Digging, weeding, planting, transplanting, or simply running hands through loose soil in a pot on a table. The physics requires contact, soil and skin, nothing more. A person over 65 with limited mobility can fill a wide, shallow container with garden soil, not sterilized potting mix which has been heat treated to eliminate the microbial diversity the immune system needs, but actual garden soil containing the full billion bacteria per gram, and place it at a comfortable height. 10 minutes of working the hands through that soil activates every mechanism described in this discussion. The container on the kitchen table provides equivalent microbial diversity, equivalent temperature differential, equivalent mechanoreceptor activation, equivalent transdermal ion exposure as the garden bed. The garden adds sunlight, bird song, and the wider sensory environment, each covered in their own discussions, but the hand-soil interface operates independently of the setting. If you garden already, if you are the person who spends weekend mornings with hands in earth and has always known that something happens but could never explain what, you now have the inventory. 6 mechanisms, 6 pathways through or across a membrane 15 micrometers thick. 6 systems in your body responding to an input profile the indoor world has entirely eliminated. The gloves you wear to keep your hands clean are blocking 5 of the 6 mechanisms. The temperature differential is eliminated. The glove insulates the skin from the soil's thermal mass. The transdermal ion exchange is blocked. The rubber or fabric prevents ionic migration entirely. The microbial seeding is blocked. No soil organisms reach the skin. The vaccae delivery is blocked. The bacterium cannot cross a synthetic barrier. The mechanoreceptor complexity is attenuated. The glove smooths the textural variation and reduces the sensory resolution of every receptor type. The only mechanism gloves partially preserve is the gross somatosensory input, and even that arrives at reduced fidelity through the barrier material. The physics operates through the membrane. The membrane must meet the earth without intermediary. 1 billion bacteria per gram. 4 mechanoreceptor types firing simultaneously. Cool earth against warm skin. Dissolved minerals migrating across a membrane 15 micrometers thick. Geosmin at close range reaching the olfactory bulb before conscious thought. Immune calibration through toll-like receptors activated by organisms the indoor world never provides. Serotonin production initiated by a bacterium that crossed the skin of the hand. 6 mechanisms. 6 pathways through a boundary thinner than a sheet of paper. 6 systems in your body responding to an input profile that the indoor world has entirely eliminated. 10 minutes. Bare hands. Soil. The body that wakes up in a garden is responding to a sensory, chemical, thermal, and microbial input profile that its immune system, its somatosensory cortex, and its brainstem have been calibrated to receive, and that the indoor world has entirely eliminated. The hands know what to do with the earth. They have been doing it for 2 million years. 17,000 receptors per hand, waiting for the signal the indoor world withheld. They are waiting to do it again.